Oh? Yes? Okay, good. I usually talk to lecture halls bigger than this without the microphone, so I'll have to tone it down a bit. It's a pleasure to be here. Every 2 years, the trip to Penn State to meet with old friends, but also to interact with the young members of our community, to really share these methods, to help you pack up your toolbox, if you will, of methods that you will be using in your careers. And so it's a great pleasure. But once again, Karsten has put me after Serena, and that is a very hard act to follow. So, but I'll try my best. Also, I guess I'm last on the program. So, after a long day and a half, everyone's tired. Right before lunch, you're hungry. So the bar is pretty high in terms of me keeping you engaged and entertained as well. So, but we'll do our best. So here we go. What I'm going to do this time is, let's see, I've got a laser pointer here. Hang on, there we go, got it. Okay. So this here, what I'm going to try to do is just give a brief overview of the method. And of course, with this community, focus on the key issues with metals. That's going to be our main focus. And just an application or two at the end just to show how this method can be useful. This is a method that's kind of quite different than most of those that you've been talking about. It doesn't fall on the energy scale. It's old. Lavoisier was doing these measurements. But I think you'll have to put your coordination chemist hat on and really start to think about the type of information you can get out of this technique known as isothermal titration calorimetry. Okay, so the method, some technical issues with regard to metals. In particular, we're going to be focusing a lot on metal buffer interactions because that's, we use buffered solutions and we've got to be aware of the issues with regard to that. But this is, I think, a wonderful, really opportunity for this method to provide very useful information, and that's proton flow coupling to binding. And just a couple of examples here. Okay, so metal ions of interest, okay. John, in his talk earlier, said, the main metal he focused on is magnesium. Okay, that's there, but this community tends to focus more on transition metals and occasionally some of the heavier metals. Have I forgotten any? Anyone want to add to this list? Okay, so I think we got most of them here. Questions of interest when we're interested in metal-protein interactions, which maybe if you're studying metal homeostasis or trafficking or metallochaperones or create, binding of metals to a metalloenzyme. You're looking at specific binding, and you're interested in stoichiometry, affinity, and this technique actually gives us the thermodynamic foundations for that type of interaction. You might worry a bit about non-specific binding and effects on proteins. We'll see that we can get around that with this method. And of course, one also might focus on metal ion contributions to protein stability. That's the technique of differential scanning calorimetry. I'm not going to touch on that today. We don't have a DSC here that you can have hands-on experience, but over in the biotechnology lab, Julia Fecco has a robotic DSC, and we'll take a field trip over there and actually see that, which is a really pretty amazing device. Okay, caution here, we're going to be interested in measuring binding constants, but we're going to be interested in measuring the enthalpy of these processes. But the caution here is these are bulk thermodynamic measurements. So everything that contributes to the enthalpy goes into what we measure. So we have to be aware of that, okay? And we'll need to consider if there's any coupled reactions or competing reactions that in fact are contributing to the signal that we're measuring. And clearly, and this point was made just in Serena's answer to my question, we want to compare and confirm the results with other methods. So we learn a lot by using multiple methods. Okay, so here's what the technique is mainly used to focus on this. There are some other uses of the titration calorimetric method, but we're really just focusing on metal binding and trying to pull out this thermodynamic information. We're directly measuring enthalpy. This is not an indirect method like using the Van 't Hoff relationship, so we're really focusing right on the heat flow. And when reaction goes so far as to learn about the heat capacity of binding. So here's the instrument in a schematic fashion. There's one, actually that's the one that we're going to be using in our tutorial later on in the next few days. This one is contained in a glove box. Obvious reasons, in this community you might want to keep oxygen out. But here's the cartoon. We have a reaction cell, we have a reference cell. We're looking at trying to maintain constant temperature in this sample cell as we're adding a titrant, and reactions are either exothermic or endothermic. And so the heat that you need to add, if it's an endothermic event, or subtract if it's an exothermic event, that's what you're tracking. Okay? So this is how we can tell whether it's the heat is flowing in or out by basically which direction the peak is pointing up or down. Most reactions are going to be exothermic, but remember, it's a bulk thermodynamic method, so a lot of things contribute to the heat that you measure, and so it may be net endothermic. Okay? So we'll get into how we kind of extract out the enthalpy that is of interest to us. Okay, so here's a cartoon of the data. Okay, each of these are little injection peaks, injections of the aliquots that we're adding. A simple titration type curve. We see here's like we would call this a binding isotherm. Okay, this is the raw data. Of course, the cartoon raw data. We'll see raw data shortly, but we'll need to take this data and integrate the area of each one of these peaks. Software will do this for us. And account for the concentrations, and now we end up with a true binding isotherm, which is kilocalories per mole of injectant versus the molar ratio. Okay? And from this we start to extract out useful information. Kind of obviously here, we have binding here initially, lots of heat. At the end we have very little heat. Okay? This is the heat of dilution. Okay? You always have to account for that because it's contributing to all the peaks here. All right? But by comparing the two, we can in fact pull out the change in enthalpy for the binding. Okay? Clearly the inflection or inflections will tell us where there's a change in the binding, and that gives us information about stoichiometry. And then, because this is an equilibrium situation, we're actually able to determine the equilibrium constant associated with this. Okay? So here, with any technique where you're measuring an equilibrium situation, you have a range over which you can measure it. And practically for this technique, it's about a 5 order of magnitude range from about log K of 3 to 8. Okay, so you can do the reciprocal and figure out what that is in KD values. But this little plot over here gives us a reason for that. This is what we call the so-called C window, if you will. All right? Clearly, if the binding is very tight, everything binds till you reach saturation, and then there's nothing. You really get a step function. All right? If the binding is very weak, not everything's binding, and it takes you large excess till you saturate. So qualitatively, you can look at the data and actually get a sense of what is the order of magnitude of my binding constant just by basically the slope of the inflection here. Okay? So, but, from this data, K gives you delta G, measure delta H, you can get from a good data set a complete thermodynamic analysis. Okay? And of course, this idea of temperature dependence then pulls out the change in heat capacity. All right, so that's kind of the essence of it. Now we're going to actually kind of deal with some of the issues, the aspects that one needs to consider when you're thinking about this data. Okay, so here's the advantages. We're going to mention just a bit about this proton competition. Each one of those injection peaks has some kinetic data associated with it. So there is an ability to extract out kinetics from these measurements, and you can even do this for redox reactions. It's a more esoteric use of the method, but that is something one can consider. Okay. All right. Now, basic uses. All right. People use the technique for many different purposes. One might be, is there binding? Do I see any heat when I mix my ligand and my protein? Okay. Next, what is binding stoichiometry? Okay. These numbers tend to be soft because there's a lot of sources of contributions of error. You got two concentrations, you got volumes and all, so these numbers are a bit soft. Okay, the binding affinity. So a lot of biochemists, when they use this method, they would actually kind of stop here. Okay, can I pull out a KD? Can I get stoichiometries? Okay? I would hope that this community would be interested in going further to learn something about the fundamental thermodynamics that's going on, because you're measuring enthalpy directly. Okay? And if you can take this experimental enthalpy and extract out of it the enthalpy associated with the exact molecular event you're interested in, then you've got some real useful information. This idea about coupled protonation or deprotonation, we'll talk about that in some detail. This, I think, is a very seen more and more use in the literature, and I think it's something that is a very wonderful contribution that this method can provide. Finally, of course, we can pull out the entropy. Clearly, if you just using ITC data, delta G and delta H are under the same conditions. But consider, you have a binding constant that you've measured through some fluorescence method, okay? And then you use the calorimetry only to determine the enthalpy. That's fine, as long as both those measurements were made under the same conditions. Okay, you got to make sure that those values are comparable. And this is kind of the more advanced methods, okay? Heat capacity. Just a plug here that perturbations. This is a wonderful method for looking at how, compare mutants, compare conditions, compare pH. So oftentimes we're looking at delta-delta thermodynamic parameters. And there's a lot of use for that. You find out what a perturbant is doing to the situation. Okay, so manipulation of the data. Okay, you start with your raw data, which is usually kind of slopey. You got to flatten the baseline. Okay, subtracting out this heat of dilution, really important step to get accurate numbers. Okay, you can do it from this extended region of the spectrum. You oftentimes run it out so you do get a good measurement of that heat of dilution. Then you need, of course, integrate the peaks very carefully. Okay, you got to make sure your peaks are well separated so that you're in fact integrating all the heat for each injection. And so then qualitative analysis, is there enough heat? Are you seeing associated with that? Well, you can change the concentration. This depends upon how much stuff you have present. Is the data within this so-called C window or not? Okay, stoichiometry. So, you get some initial data, better data, optimal data, reproducible data. Where does publishable data fall on this continuum? Okay? I mean, I hope it's out here, but, it might be closer to the beginning here. But this is a technique that does take some working with it that you can actually get some very good reproducible information out of it. The errors are enough that you need that reproducibility to get numbers with good confidence and low error bars associated with it. Okay, now you've noticed that these data points often have lines fit through them, okay? They're fit to lines, okay? Some information you can glean directly from the data, like the stoichiometry, but in particular, the fitting of the binding constant really requires, a model that fits it. So now you have to put your thinking caps on and say, okay, what's going on here? Is it a single site binding event? Can I use the equilibrium relationships for that and a model that fits it to pull out these parameters? If you have more than one binding event, okay, there now you have to worry a bit about how close are the two affinities, okay? Are they the same? Are they different? Do I use a sequential binding model, or do I use a model where I have two independent sites so their binding is independent? Finally, sometimes you can set up measurements where you do competitions, right? And we'll see a mention of about that a bit later, but where the binding of one thing and something else displaces or competes it, and so you can pull out the unknowns associated with that. In the end, though, the bottom line is you get some good condition-dependent values, and now you have to do what we might call post hoc analysis to pull out condition-independent values. The data, you want it to be independent of the buffer, independent of the specific conditions, and that's going to be a key thing. Okay, dirty secrets, okay? I guess every method has, kind of, here's the trouble with it. Here's, Serena alluded to maybe some people that haven't done such a great job with their fitting of their X-ray absorption. So here's the problem with this method, okay? The instruments aren't that expensive. You know, there's probably one at your college or university, okay? But some people have been frustrated with their ability to collect data, and you know, sometimes, you know, with regard to the numbers that are reported from this data, there are clearly some problems with it, okay? For those that were here 2 years ago, I actually crossed over a line, which I feel badly, but it never came back to bite me, okay? I presented some data from a paper I was reviewing, okay? Now, is Kara Bren here? Okay, Kara was in the audience, and I saw her kind of grimaced when I showed this data. She said, yeah, that's confidential data, but it was a clear teaching moment, so I, in fact, used that, okay? And I hope everyone learned from it, and I never got feedback from the editor about it, but anyways, okay. But here's the problem. The instruments are very sensitive. They're prone to contamination, so care and cleanliness, you want to make sure that, you know, the instrument is handled very, very lovingly, okay? Competing reactions and processes. This is a big thing. You have to account for them or eliminate them, or account for them. And here's the bottom line, which is our lead-in. There are more complications when metal ions are involved. So let's talk about those complications for a few minutes. Okay, so metal buffer interactions. Metals are Lewis acids, buffers are Lewis bases. You put a metal in a buffered solution, you're going to form, there's some interaction. It may be weak, but it may be strong. We'll see some cases of very strong metal buffer interaction. We know metals do redox chemistry, hydrolysis reactions. Just putting a metal ion in solution, particularly the stronger the Lewis acid, you know, you get this hydroxides formed, okay? And solubility is an issue. Finally, this idea that metals, when they bind to proteins, they're oftentimes in competition with protons. Where does the proton go? Well, it gets bound to the buffer. Okay? That's a heat associated with this step here that has really nothing to do with the metal binding to the protein. So let's look at these and think about how we might either suppress them, eliminate them, or account for them in our analysis. So first of all, these things here that are really competing, okay, oxidation reduction reactions. So again, it's knowing when to expect it. If you're studying copper 1, you're going to have to worry about making sure it remains copper 1. You also, with copper 1, have to worry about disproportionation reactions. So if anyone's worked with that, okay. Other metals, it's not a problem. Anaerobic conditions, of course, is the way to go with that. You might think, well, I'll just put in a reductant. Put in some DTT, put in some TSEP, right? But if you're doing the titration and oxygen's leaking in and it's reacting with the TSEP, there's a heat associated with that, okay? So you have to worry a little bit about it. In fact, these are non-innocent things, you know. Metals bind to DTT. So anaerobic conditions are the way to go to suppress this if possible. Precipitation reactions, you know, it's a non-destructive method. You can pull out the sample afterwards and look to see whether you've got gunk in there or not, okay? Usually when that happens, you can't get any data. Data is awful. So, but you can avoid precipitation by adjusting the conditions, common ones. Also, inorganic chemists will tell you avoid phosphates with metal ions. Metal phosphates have low solubility. We try to avoid phosphate as a buffer with this method to avoid that, okay? Reactions with water, this hydrolysis. Oftentimes that chemistry is concentration dependent. Remember, the sample in whatever's in the syringe is more concentrated because you're adding small aliquots. And so its concentration changes dramatically from initially to the final state, okay? So one way to at least identify that is let's run the reaction in the reverse direction, okay? If you're possible, you can just switch the identities. If the metal's in the cell, its concentration isn't changing at all. You're titrating in the protein, okay? But, and this is maybe the bottom line is the key one here, which actually accounts certainly for both of these phenomena. Deliver the metal as a well-defined complex with a ligand, or even the buffer can account for that. So these are, in fact, tools of tricks that are very important for us. All right, let's go back to these two here, metal buffer interaction and this idea about proton competition or proton displacement, okay? So again, metals are Lewis acids. They compete with protons, which are also Lewis acids, okay? Coupled equilibria are very common, and we need to account for the heat, particularly for protons that are displaced from the protein or protons that may bind to the protein when your metal binds. May seem kind of unusual, but there are cases of that. How about the buffer, okay? Interaction with the metal ion, it can suppress these side reactions. You know, it may be help out in other ways. And it can provide a competing ligand for the metal ion. We'll see that in just a minute when we, but you need to know the metal buffer stability and the enthalpy to account for that. Interaction with protons, data in different buffers can quantify this, and we'll examine that. Here's an interesting one. Let's say your metal binds to the protein, displaces a proton or two or more. It binds to the buffer, there's a heat of protonation. Depending on the buffer, that can be a large or small contribution, okay? And other species in solution might be of interest. Okay. So how does one account for all that? You use thermodynamic cycles, Hess's law. This goes back to freshman chemistry. It's very, very, because we're dealing with condition-independent variables, okay? In fact, we can make up these thermodynamic cycles. Here's what we're measuring. Overall, you know, metal, some bound with the buffer or not, the ligand or protein, different protonation states. In fact, we end up with this. But this is what's of interest here. Right? So how do we do that? We account for each of these individual equilibria and subtract out and get out the one of interest. So we do this quite routinely, and it's a way to get to the actual number that, from your experimental value, the number that really does relate to what you claim it is. Okay? Now, interesting point here, you know, if protons are displaced, okay, from the protein, they bind to the buffer, okay, but the number of protons depends upon how many protons. So there's a clue there, okay, that you can use this data to actually count protons or quantify them. Okay, metal buffer interactions. This is in fact data that sometimes is available, sometimes isn't. But it's some data that you need for this post hoc analysis. Just to give you an example of how one might obtain this and actually point out that this metal buffer interactions or binding can be very significant. Let's look at this way that we actually measured the interaction between copper-2 and a buffer bis-tris. Probably everyone's used bis-tris in one form or another. Okay? So what we do is consider a copper ligand complex that's pretty well characterized. And these are two examples, TEPA, tetraethylene pentaamine, or this one, DTPA. You know, the copper ligand interaction is pretty well known. NIST has databases of them. In fact, look at the stability here. It's pretty high. Okay? These are very stable complexes. Okay? So what we're going to do is we're going to do one of these ITC titrations, okay, where we're titrating the ligand into the copper solution, but both solutions have this bis-tris ligand present in fairly high concentration. So what we have here is a competition between the buffer and the ligand. And in fact, you know, this value is way outside our C window, right? That would be a step function. But look at the data that we see. That's nicely fit. Indicates there that in fact copper with bis-tris has a fairly stable complex. It's competing with the ligand. And so by looking at the difference between the numbers you get out of this type of data and the numbers that have been reported for the copper with this ligand, we can extract out the metal buffer interaction. So for example, here's TEPA and DTPA, here's bis-tris. Log beta values, 10 to the 12th. I mean, that's a pretty stable complex. Copper's an amines. It's an amine type ligand. With DTPA, we get a number that's fairly similar. You know, we look at the average of these two, and so that's what we're going to use for our copper bis-tris stability constant. We're going to pull out the enthalpy from that because we do know the copper TEPA enthalpy. And again, we're looking at an average of those two to get a value that can be associated with that. So you're getting a sense here about, the errors that are associated with these types of measurements. It's tenths of kilocalories, but the values are calibrated with regard to that. Okay, so in fact, here's data for a whole bunch of these amine buffers. You notice they all form fairly stable complexes with copper. All right, let's see. Data in different buffers can quantify displaced protons. So I'm kind of on the road to sell this as one of the main values of this technique of isothermal titration calorimetry. So first of all, just a quick example of how this buffer protonation can be a big deal. So here's some of our early data we collected, just simple amino acid histidine binding nickel. Okay, same measurement. We're titrating histidine into nickel in Heaps buffer and phosphate buffer. The concentrations are a bit different, the pH is a bit different, but look at this data. Net exothermic, net endothermic. Okay, wow. I didn't believe the data when I first saw it. I said, you must have gotten something wrong, but in fact, what's going on here, think about it. It's an amino acid, so the amine is protonated. Copper's going to bind, okay, displace that proton. Proton binds to the buffer. Turns out the enthalpy of protonation of Heaps is about 4 times that of phosphate. So this signal is exothermic because of the protonation of Heaps that's coupled to the binding. And so the phosphate has very little contribution, so it turns out to be net endothermic. So that's kind of a clue that this buffer protonation contribution can be a pretty significant contribution to your measured enthalpy. So how does one do with that in a quantitative sort of way, to count protons, if you will? So here's some data from a measurement study we did of a peptide, a sequence from a protein that's involved in iron transport. And this sequence jumps right out at you. It's histidine glycine repeats that are flanked by prolines. It's like, why did nature put that in that protein? There must be some coordination chemistry associated with that. An inorganic chemist might have designed that peptide for metal binding. So we were interested in looking at metals binding to it. And here's the data for zinc binding to this small peptide. Okay, so now without looking at the data over here, numbers over here, look at the data. What do you think? Weak binding or tight binding? A step function? No, it's really kind of weak. In fact, it's about, 100 micromolar binding. Okay, but the key thing is the enthalpy that you extract out of this. So remember, this is the enthalpy for the overall enthalpy associated with the binding of copper to this protein. And that's what you measure here, okay? And here's all the other contributions. This is our Hess's law type of analysis. So let's take that and rearrange that equation. We're going to, now when you change the buffer, you change 2 things. Not only do you change this protonation of the buffer, but you change the metal-buffer interaction. Now, if we know that, we can, in fact, conclude that over here with our experimental value. And if we do a simple plot of that, which is on this axis, versus the buffer protonation enthalpy, the slope is, in fact, the number of protons. So here's data in 4 different buffers, pretty good R² there. Slope is about 0.8. So, in fact, we know that when copper binds to this peptide at pH 7.25, that about 0.8 protons are displaced. Okay, that's a useful piece of information if you're trying to understand the coordination chemistry. And so that type of plot becomes pretty useful to quantify protonation coupled with binding. Okay, so those of you doing the ITC module tutorial here will be looking at copper binding to albumin. Okay, here's albumin. Turns out there's residue number 4. Disorder here, at least in these early structures, okay? And those 4 residues, in fact, include a histidine in the 3rd position. So it's that N-terminal binding site, which is a well-known motif now, that binds copper and nickel in this strong square planar coordination. Okay? If we look at the data for this binding by a variety of different methods, ultrafiltration, dialysis, even ionoselective electrode, some case in different buffers, different pHs, sometimes with competing ligands, we notice that the log K at pH 7.4 is generally pretty high. Okay, So how are we going to put that within our C window to get some experimental data? Well, we're going to have to set up a competition. And luckily we know that some of these amine buffers bind copper fairly tightly. So by using a buffer like tris, we in fact can set up a competition between the tris and the bovine serum albumin to actually measure that. So here's data that, much like you will be collecting when you actually load the instrument and see your own data. This is a little bit higher pH, but simple one-site binding of copper binding to bovine serum albumin. From this we can pull out the enthalpy in tris at this pH, okay, and get a sense of the binding constant out of that. We'll analyze that data. Okay, one last thing about kind of experimental design. So I already mentioned once this idea about maybe doing a reverse titration to in fact try to understand what's going on with regard to, well, to avoid precipitation, if you will, or solubility issues. Okay? But forward and reverse titrations are pretty interesting because, remember, one species starts out, fairly dilute, okay, and some starts out very concentrated. And so the two directions, in fact, have different titration shapes. And by fitting both the forward and reverse, you gain extra confidence that your fitting model is in fact accurately determining that. Displacement titration. So this could be kind of interesting. If you have a binding site, you're looking at one metal and you're looking at the competition, so you figure out the affinity of the second metal. So here we're going to titrate one into a previously metal-bound protein. This is the equilibrium of interest. And of course you're going to get a binding if the affinity for the second one is higher than the first. Now, thinking about this, what if the enthalpy associated with the binding of metal 1 is the same as that for metal 2? Oh, you might not have much heat or very negligible heat. In other words, you're measuring enthalpy, and that is direct. A lot of it that directly relates to the metal ligand bonding, okay? And so that would be a reason why you would in fact measure a difference in enthalpy here as a way to probe that. Chelation titrations. Okay, this is again a competition type experiment where you'll start with the metal-bound protein, you titrate in some ligand, some chelate like EDTA or something that binds the metal with a higher affinity than the protein, and then you can pull that out. This might be useful, in fact it proved useful to us when the protein by itself had stability issues. So you preform the metal-protein complex and you can kind of get the data in a reverse sort of way. Okay? So these are all useful types of things. Okay, so now just to wrap up with just a couple of examples. The first one, I'm not even going to show you any data, but the first one is a case of zinc binding to carbonic anhydrase. So carbonic anhydrase, one of the better studied, I don't know if it's the hydrogen atom of the bioinorganic world, but it's pretty well studied. And early on, Eric Toon and Carol Ferke, Carol has worked on this for quite many years, published a very nice study, thermodynamics of metal ion binding to carbonic anhydrase. And they did a lot of very careful measurements, okay? But this was 2001, and since then is when a lot of this understanding of all of this, of careful analysis of the data has emerged. And so a few years, about a decade later, Joe Emerson at Mississippi State with his colleague Ed Lewis, who by the way is one of the grand old men of biophysical chemistry, okay, did a study where they revisited zinc coordination, taking into account the types of analysis that I've described to you here. It turns out that this group found that the affinity was like 3 orders of magnitude lower, and in fact, the binding thermodynamically was somewhat different than the original measurements. So this is maybe a lesson of careful analysis. What I'll do on the Dropbox is actually upload these 2 papers and then kind of a description of kind of how we have become more sophisticated and accurate in our analysis of ITC data, using this as an example. I don't think Eric I know Joe would be happy about that. I don't think Eric would mind because we've come a long way in understanding of this method. Second example that I wanted to mention is from some of our own work, a short little story that has to do with a phosphodiesterase. Now, this is glycerophosphodiesterase from Enterobacter aerogenes. This is a member of a metallohydrolase family that uses two metals, okay? But it's an enzyme that has, in the resting state, only has a single metal bound. So the second metal presumably comes in with the substrate when it's binding. So metal binding is important for the catalytic activity of this. Here's a crystal structure from David Olis at the Australian National University. I think his group might have even been the ones that found this. This is a crystal structure with an excess of metal. I forget which metal it is, if it's cobalt or manganese, but you can see both metals bound. So in this particular form, this is, I think, something from the crystallization conditions. But here's a cartoon of the active site. There's an A site and a B site. The A site is the site where the metal is bound more tightly in the native resting form. In this collaboration with Gary Schenk at Queensland, Gary's group prepared a couple of variants. Now notice in this variant, at the B site, they took the asparagine and changed it to an aspartic acid. So an amide to a carboxylate. Okay, so there's a first coordination sphere variant. In this one, they took the serine that's hydrogen bonded to this asparagine and changed that to an alanine. So they eliminated a second sphere interaction. Okay, and this one, in fact, retains high activity. So this variant, in fact, has similar activity to the wild type. So in collaborating with them, basically what we did was looked at manganese and cobalt binding. And here's the ITC data for the binding. You can see, not such tight binding, maybe a little bit tighter binding. We're kind of picking up more of a sigmoidal shape here with the cobalt. But when you do the binding, and look at the stoichiometry, it's 1 for each one, okay? So under these conditions, you're seeing the binding of the first metal that's bound in the resting enzyme. But when you bind these metals with phosphate, now think about a phosphodiesterase, okay? Phosphate is a product, right? So this is an enzyme product complex, if you will. What you find, in fact, is a much more complex binding. Isotherm 2 binding events, okay, for both the manganese and the cobalt. So here we're kind of able to kind of pull out the thermodynamics of the metals binding to this protein. The second metal binding with the substrate, in fact, is pretty important catalytically. Okay, now, I've tried to convince you that this idea about varying the buffer so you can quantify protons is useful. And hopefully this makes that point. So when you look at this ITC data, okay, and you use plots like this, where we're looking at the enthalpy of binding versus the buffer ionization with a variety of buffers, tris, tes, heps, mes, pipes, a range of enthalpies of protonation, we end up with these nice plots where we can extract the number of protons. So these two data sets are for the metal binding in the alpha site, the first one. This data set is for the metal binding in the beta site when the phosphate binds. And this is quantified as about 1 proton, and this very little. So when you think about a metallohydrolase, we oftentimes think about one of the roles of the metal is to stabilize a deprotonated water, hydroxide. So when you form this site, you lose 1 proton. And that proton is actually in the binding of the first metal. You don't need the second metal to stabilize that hydroxide. Okay? And here's the data that, in fact, is the thermodynamic data that shows how that there's only a loss of proton with 1 metal binding, and it's the first one, okay? And that the phosphate analog or inhibitor increases affinity at both sites. There's an entropic benefit at the alpha site, okay? So in fact, notice it becomes more entropically favorable, okay? And at the beta site, all you have is the enthalpy associated with that. So we're understanding kind of the thermodynamics of the two metals binding. Now, what about these variants? Okay, again, deltas are useful. So here is our asparagine aspartic, the first coordination sphere variant, okay? And here's our second coordination sphere variant. No phosphate now. Okay? We add the metal, and the data shows, one, we get 2 binding events. So now we've kind of beefed up that beta site, so now both metals are bound. We have a fully active protein even in the resting state. We don't have to add any extra metals. And what's the data saying? Well, basically, it's saying that, in fact, here's the thermodynamics of the alpha of the 2 sites. So clearly, by adjusting both the first coordination sphere or the second, you're, in fact, proving that. And, in fact, you can see that, in fact, this is predominantly an enthalpically favored binding event by both these modifications of the protein structure. Okay, so mechanistic insight. Now, that's always, where we're kind of going with all of this, right? So apoprotein, waiting for metals. First cobalt binds, and it stabilizes hydroxide. So you think, oh, wow, the site's ready to go. Okay, but it's not active until if you add substrate, you get no reaction. If you add excess cobalt, you get both sites bound, as was shown in the crystal structure. But it's when you bind the second cobalt with the substrate that now you, in fact, find the second cobalt is adding something to the metal substrate complex that allows the catalytic activity. It's not assisting in deprotonation, because we showed that was in this first step by quantifying the number of protons. Only 1 proton, and it's in this first step. Okay, so that wraps me up here. This is some of my earlier group members who've gone on to great careers. Molly and George were here at the very first 4 years ago. Michael came 2 years ago to help with the tutorials. Caitlin, where's Caitlin? There's Caitlin is here to help with the tutorial. Matthew, back in the corner, are here to help with the ITC tutorial. And I welcome you to come and try your hands at collecting some of this data. And I'd be happy to take any questions.